I'm excited to be able to present our paper. I watched a lot of the previous journal clubs and I was really impressed by the work which was reported on there. So I'm really excited about that. I was told that I shouldn't do a lot of background, but I just wanted to get you on the train of thought we had for this analysis. So as you know, stress and depression impacts brain structure. And as you see, there's a number, as in humans, there are a number of regions which were shown to be smaller in depressed patients compared to healthy volunteers. And this was also shown in post-mortem studies. And for ketamine, as you may know, ketamine is able to restore these deficits in dendritic spines, for example. And this is a very nice study by Moda-Sawa et al., which was published in 2019, which shows that chronic stress leads to dendritic spine loss, which can be restored by a single dosage of ketamine when investigated 24 hours after ketamine application. And this is also mirrored by difference in behavioral parameters such as immobility, as you can see here. Neuroplasticity is a major mechanism of action which was shown for ketamine, which was shown in a number of studies, mainly preclinical studies. What really inspired us to do this work I will show you today is a study by Treccani et al., also published in 2019, which showed that s-ketamine can reverse dendritic spine deficits in a very short time period of 1 hour within hippocampal CA1 region. They used a genetic model of depression in rats and had a control condition of non-depressed rats. And they used a single dosage of s-ketamine and then checked for parameters of synaptogenesis and neuroplasticity 1 hour after application. And they saw significant increase in spine density in mushroom spines and long thin spines. But here it has to be noted that they found this effect only in the animal model of depression, but not in the other animals. But there are also other studies which could show such or similar effects in wild type mice, such as Lee et al. 2010. So, based on, we were then already interested in the hippocampus and to check hippocampus volume within our study sample. And then I was thinking about what could we also include, which other parameters could be relevant. There is a larger body of evidence which showed that the BDNF Val66 genotype, which is a major, which is a SNP, a single nucleotide polymorphism within the BDNF gene, which is very relevant for the action of ketamine also. That the genotype influences hippocampal volumes in healthy volunteers. This was shown in a meta-analysis in 2012. And that the MET carriers tend to have smaller hippocampal volumes. And the MET allele is kind of a risk allele. And it was also shown for ketamine that MET carrier have a decreased antidepressant response to ketamine, which is also reflected by animal studies, which showed that MET knockout mice have an impaired ketamine-induced synaptogenesis. So to kind of translate this information I showed you into our study, we wanted to investigate the hippocampus and hippocampal subfields. I will show them later on, and check for the effect of ketamine on two hippocampal subfields and the influence of the BDNF by 66-MET genotype. So this is the study design we used. We had a double-blind crossover design. So all participants completed 2 MRI sessions within 1 to 2, maximum of 2 weeks. And within each session, they had 2 structural MRI scans, one before administration of ketamine. Then there was an fMRI part, which is not part of this paper. And then they had intravenous application of ketamine over a time period of 20 minutes. First, they had a bolus of 0.11 milligram per kilogram, and then followed by a maintenance infusion of 0.12 milligram per kilogram per hour over a time period of 19 minutes. This resulted in a dosage of about 15.5 milligrams per participant. And this was done within the scanner. And after 1 week, they had the same scanning procedure again, but received the crossover agent, placebo or ketamine. And for the BDNF SNPs, we pooled the MET allele carriers within one group. There were 12 MET carriers within our sample, and the VAL-VAL within another group. So the MET carriers would be the risk group, and the VAL-VAL the non-risk group. And this resulted in kind of very small samples, but kind of same size. Here you see the main results. Here I can show you the hippocampal subfields. We did the analysis with the free surface software, which is an automated segmentation software, which does an automated segmentation of all these hippocampal subfields. As I showed you before, the Tricani paper showed these differences within the CA1 region, which is the red part here. We did linear mixed models in order to compare placebo and ketamine. And using this model and adjusting for sex, age, and total intracranial volume, we found significantly larger hippocampal subfields in the ketamine compared to the placebo condition. But we couldn't attribute these effects to a single subfield. It was kind of an overall effect. The highest p-values were shown for the CA1 region left, which is kind of nice because it reflects the results which were shown for animals in the Tricani study. Unfortunately, we couldn't show any effects of the BDNF genotype onto our results. This may be due to too small sample size, for example. Here again, you see the mixed model estimates just for visual representation. Here you see the CA1 region, and you see that the effects are not very strong, but strong enough to be significant. When we checked if there is an effect of ketamine onto absolute volumes of the hippocampus, we couldn't find significant differences, as you can see here and here. Here you see that the total volumes remain quite stable, and we can see only when using this more sophisticated approach and the subfield segmentation, we can see these differences I showed you before. So to sum up, we can show that ketamine has a rapid effect on hippocampal subfields within the time period of 1 hour, which is a very short time period in humans, which I think is a very nice starting point to go further into this field of research, because there is a large body of evidence within animals and even on a more basic research level. But until now, there's only very little information about this, or little evidence in humans. So what would be really interesting, of course, is to now use this approach in a sample of depressive patients. I think this should be the natural next step. Of course, we did only one MRI after the application of ketamine. Maybe effects would be even stronger when doing serial MRI scans, and maybe after a couple of hours later, we would see even more stronger effect, but we didn't check for this. So when thinking about translational studies, what limits us as human researchers is, of course, that we only have indirect measures for a lot of stuff which can be done in animals. And even MRI is kind of an indirect measure, of course, of synaptogenesis or of other neuroplastic processes. So, of course, MRI signal is prone to other factors, such as, for example, fluid changes. Also maybe the influence of infusion, as in our case. Also glia can influence the signal. So we are, of course, not that sure about what we measure as in animals, but there are some studies showing that this is again an animal study, but there are studies showing that gray matter loss in MRI is due to loss of dendrites and synapses, which gives us kind of confidence that what we measure might be the right thing. Another problem, or what we could maybe discuss later on, is that the translation of the dosage onto humans is also not that simple, because we use intravenous infusion in humans, but of course in animal studies often intraperitoneal administration is used, and there is not like a rule of thumb of or standard how to translate this. It was shown that the bioavailability after intravenous infusion is higher than after intraperitoneal administration, and that plasma concentration after IV bolus is higher than all of the other routes of administration. But unfortunately, the second study did not directly compare IP versus IV. So we cannot really say if the dosage we used is comparable to the one used in the study, for example, by Treccani et al. So the conclusion would be, we did find first evidence that ketamine has rapid effects on hippocampal subfield volumes in humans, but more research is needed in order to extend these results onto other population, especially depressed patients, to check for the optimum time point when to study these effects. And to maybe also translate this onto functional parameters of within MRI setting and so on. So this would be short overview from my point, and thank you for your attention.
Thank you very much, and very, very interesting and translational work. Wasn't it? We already have some questions that already kicked in, so maybe I can start reading some of them to you. So Tiago was asking if you can talk a bit more about why you chose the dosage, the 0.2 milligram per kilogram administered over 20 minutes, and not the typical, more famous 0.5 over 40 minutes.
Yeah. First of all, we used s-ketamine, and I think most studies use like the racemate, so the 0.5, I would guess, is for the racemic ketamine. And as you saw, we had a very long scanning period of 90 minutes, and administration was done within the scanner. So we had a pilot study where we tried to find an optimum dosage, where we could see like psychotomimetic effects within this sample, but without people jumping out of the scanner, so to say. And at the time, there was this 0.5 was not written down at the time we started with the study. So we hadn't this kind of input. So this is through the pilot study, I would say we chose this special dosage, but it's rather low, and we are aware that it's rather low, but we had like strong effects on our fMRI data and on psychological parameters, and also now on this data we have now. So we're happy with it.
And when you say that they had these psychotomimetic effect without jumping out scanning, how did you, like, meaning like from what you saw from the scanning, or just like through self-reports? How did you establish that?
Yeah. We used the PANS because, as I told you before, at first place, we wanted to use it as a model for schizophrenia. So we used the PANS. And we used also the altered state of consciousness scale, which is a scale to measure this, and a general psychiatric screening instrument. So this is what we used before and after scanning. Yeah.
And Vanessa was asking if you think that racemic ketamine would induce a different effect on hippocampal subfield volumes. And did you find any difference between male and females?
Yeah, we didn't. Of course, we didn't have this large group of participants. We only have 31, so we couldn't. We didn't really compare male and females. We didn't find an effect of sex on our results. We used this as a confounding factor in the model, so we didn't find a difference. But I guess. I think in another journal club, there was kind of data on estrogen and so on. So it would be really interesting, but we didn't check for it. So, yeah.
Simon was asking, you just mentioned in terms of the fMRI setting, do you think functional connectivity might be a more meaningful measure in terms of how ketamine is affecting healthy volunteers and their brain structures?
No, I think, yeah. There are some studies, and we also published studies that there is an effect on connectivity and also on just bold signal, whole brain bold signal. I would think to do both within one setting and to check like maybe connectivity of the hippocampus between in a larger group of participants, because I think then it would be good to have a larger sample. Would be interesting to combine both structural and functional data within one study. Yeah, this would be nice, I think.
Any correlation with dissociation in your findings?
No. Okay. No.
And any were like all subject, did they respond to ketamine?
No, that's what we were talking about earlier on. It was really, this is something which I find really, really interesting. I don't know, maybe we can discuss it later. That this is so variable. I didn't expect it when I started off with this, but there is really a large variance in this. Some people are just really, really psychotic, or they have dissociative phenomena, have anxiety, or very euphoric, kind of react very strongly. And some people said, I guess this was the placebo. And it wasn't. So there's really, really a large difference. And I don't know if it's like relevant in a medical or psychiatric context, but I think this has to be something. Why is this? I think it's really interesting. And why is this the case? Is it like something pharmacodynamic? Is there a difference? Would people who react very strongly have a better effect also from an antidepressant point of view, or is it just not meaningful and kind of noise within? I don't know. So I think it's really interesting.
Well, that's I would say definitely an open question still for the field as well. But it seems like that you didn't detect any association in your sample, right?
No,
but when we talk about
check for this.
And when you talk about response to ketamine, that's what you mean, like the sort of dissociation during the infusion. Yeah. Or did you also have any measure in terms of like change in their affect, for example?
Yeah. We had the brief psychiatric rating scale, which also measures like mood, but we didn't find any difference before and after ketamine for healthy volunteers. So, yeah.
We have a question from Meng Li. How could we exclude the influence of cerebral blood volume effect for the volume change in hippocampus?
I don't know. The problem I thought when I started off with structural MRI, I thought, okay, this, you see like kind of a picture. This was when I was starting off with it, and okay, this is like reality. But of course, the MRI signal is very prone to influence of cerebral blood flow. So we tried to do our placebo condition was an infusion of saline with the same velocity of infusion in order to check for this and not give only infusion in the ketamine condition. So we tried to check for this and make it as similar as possible. But of course, I don't know how you would do it.
And I think I missed a question from Rupert. Saying there was no effect in volume. So was the significant effect on the shape, or was it on the cross section? I think he's referring to a graph specifically that you shown. Maybe yeah. Rupert was the one between right and left hippocampus, maybe.
Is this one? Yeah.
Yeah, that was what I was referring to. So you were saying that there was no difference in the
overall volume, yeah.
The overall volume, but I thought that I had sort of understood you to say that there was no difference in the subfields, but there was an overall difference in the volume, and that's where I got muddled.
No. We didn't find, like for the standard, if we did only the standard analysis, which would be this, we didn't get the effect. We had it was only in the subfield segment, after the subfield segmentation, we found this effect.
So
more fine-grained, I think.
It's a different sort of analysis. The subfield analysis is a different sort of analysis, but it includes all the subfields, and it's only when you include all the subfields.
Yeah, it includes all the subfields, but it's like more just more sophisticated, I would say, analysis of segmentation. And if you concentrate on the hippocampus only, as we did, I think it's a better possibility to analyze it.